Hello and welcome to Chapter 6, An Introduction to Metabolism for EFB103. This chapter will be done in one part, so there'll just be this one video. All right, we can start with metabolism basics. Here's just a little cartoon in the background here showing some of the interconnectivity of different metabolic pathways. This is somewhat simplified. Things can get fairly complex, but I want to emphasize that here, and then we'll circle back to this, is this idea that pathways are interconnected. We're going to start to look at them one at a time, but remember all of what's happening in a cell, all the chemical reactions, that is metabolism, all of it, every chemical reaction. And so sometimes people will think of metabolism as sort of like the burning of compounds, quote unquote, burning, breaking down. But that breaking down process is catabolism. Kata means like throw. Think of like a catapult. So throwing down is catabolism, so the breaking down. And then anabolism or anabolism is the building up of compounds from smaller things. Another part of that is that catabolism produces energy, where anabolism takes energy to build things. All right, a lot of metabolic pathways occur in pathways. As we saw with, say, cell signaling, that's not a metabolic pathway per se, but there were multiple steps. And we see a similar thing with metabolic pathways in the sense there's multiple steps. So in this example, there's an enzyme responsible for converting compound A to B. That takes enzyme number 1. And then enzyme 2 converts B to C and so forth. And what this allows for is different compounds to feed in at this different point. So you don't have to start with A. You could start with B that you got from your diet, for example. Or the pathways can be regulated at multiple places. So enzyme 1 can be shut down, and so you don't get to D, but you don't have to necessarily shut off enzyme 1 and 3. So there's multiple places this can be regulated. All right, so let's think of just really generally about, energy in living systems. So energy, the definition of that is the capacity to do work. And we can split energy into two kinds, kinetic and potential energy. So kinetic, think motion, kinesis, and potential energy is stored energy. And we'll come back to these terms as we go through the next couple of weeks here. Energy can take many forms, so mechanical, electrical, heat, light, and we'll see how chemical energy is going to come up a lot in this class. Light energy is going to be important when we get into photosynthesis. Heat is always a part of this equation. And this leads us into an important concept, which is the first law of thermodynamics. And the gist of this is that energy cannot be created or destroyed. Energy can be moved around and transformed from one form to the other. And this is also known as the principle of conservation of energy. So if you think of it, the amount of energy in the universe is constant. It can be moved around in different forms, but energy isn't just created out of nothing. We'll get into this with photosynthesis. For example, here's sunlight energy, so light energy, being converted to chemical energy. So two different forms of energy, and this process known as photosynthesis converts one to the other. Now, thinking about other kinds of organisms that don't, say, photosynthesize, like us, we cannot just create energy. All the energy we get has to come from things that we eat. So here's this bear eating a fish, chemical energy in the form of food. Say this would be the same for us. We cannot create the energy. We have to acquire it from outside. All right, this is important and will become important when we get into both photosynthesis and cell respiration. One of the ways living things move energy around is through the movement of or exchange of electrons from one compound to another. And this is called a redox reaction. So in this case, oxidation is a loss of an electron. So we're seeing this one atom. This could be a bigger compound as well. It's losing an electron, so that's being oxidized. So it's lost that electron. Meanwhile, that electron has to go somewhere, so it is being picked up by this other atom in this case, so it could be a bigger compound, and that's being reduced. So that's reduction, a gain of electrons. These things happen together, so collectively these are called redox reactions because they're coupled. So this movement of electrons is important. Thinking again a little bit about electrons, they may be passed from one atom to another, but even within an atom or compound, remember, think back to earlier chapters, electrons can absorb energy and move to higher orbitals, and then when they move down to lower orbitals, they release energy. And so these are concepts that we're going to see a lot, so make sure you have a good handle on this. All right, here's a question for you. What's the smallest closed system? Given your choices here. And closed system is nothing comes in or out. All right, I like this one because you maybe initially are thinking small, right? So you may be picking one of the smallest things. But if you look at all the other choices, besides the universe, they're all open systems. The Earth is not a closed system. Sunlight comes in, right? Your cells aren't closed systems. We just talked about membrane transport. Things come in and out of your cells. So these are not closed systems. So living things are not closed systems. And that's important here when we get into the second law of thermodynamics. And there's this sort of concept. You might think of the second law of thermodynamics as the second bullet here. Disorder is more likely than order. There's a tendency toward disorder. So every energy transfer increases the entropy in the universe, or disorder. So this sort of decay or tendency toward disorder is spontaneous. It occurs without energy input or investment. And then that increased the total disorder in the universe. And so when think back to that first law of thermodynamics, when any transfer or transformation of energy occurs, something is typically lost as heat, and that increases the disorder in the universe. So if we go to other processes that are, say, non-spontaneous, that require energy, building things, those things that are built look obviously less disordered, right? So this seems to go against this whole concept of tendency toward disorder, but that still produces heat at least, and some of that energy then it contributes to the universe's disorder. So even if we have islands of local order, you still have an overall increase in disorder. And that's very relevant when we think of living things, because living things, we are highly ordered, how things are put together in a very coordinated way. But again, overall, the entire disorder or entropy, as we call it, increases in the universe for every ordered thing that there is. So let's take what we've learned now and put it in the context of free energy and stability. This relates back to potential energy as well. And I like to start with this middle one, diffusion, in panel B here, where we have, say, a concentrated dye. And as we learned before, when we talked about cell membranes, this is going to diffuse, right? So it's going to move down its concentration gradient. You can almost think of this as having potential energy. And so this has a greater capacity to do work, but it's less stable, we see here. Because as we know, because of diffusion, these things are going to spread out to a point where they are more stable, but they have less capacity to do work and a lower G, which is the free energy, less free energy. Let's go to the diving board example. You can think of this like potential energy. You're up on the top of the diving board, down you go because of gravity. And now again, you have less capacity to do work in that once you go down. Then we can apply this to chemical energy. So let's take glucose here. That's what you're seeing in this three-dimensional model here. And we can break that down into carbon dioxide and water. And our products have a lower free energy. They are more stable in this form, but less capacity to do work. So glucose, again, think of sugar as sort of a good energy source. It's because that compound has a higher free energy. So let's take that free energy idea and put it onto our different kinds of reactions. So we can start with these breaking down type reactions. So think catabolic reactions. So our reactants have a higher free energy and the products have a lower free energy. And so through that reaction, that breakdown of those products, we release some energy here. Again, what can that be used for? Maybe to do work of the cell. And that we would say has a negative delta G. So the change in free energy is negative from higher to lower. And then the opposite here are endergonic reactions. So the first ones were exergonic. Think exit, sort of giving out energy. Endergonic, you got to put in energy. So our reactants have a lower free energy to start with, and we have to plug in some energy to create the products. And so this would have a positive change in free energy. And again, this goes back to catabolism, anabolism, those broad metabolic categories where we're either breaking things down or building them, releasing energy, or investing energy. All right, so let's move on. How about this idea? Chemical equilibrium. Are most reactions in living things at equilibrium? So no, chemical reactions typically aren't at equilibrium. And you can think of this more broadly. Some might be, but if you look at metabolism as a whole, it's definitely not at equilibrium. There's lots of inputs, more products, more reactants than products, and vice versa. And so we're not at equilibrium. And I'll show you why that wouldn't work. So equilibrium and its relationship to metabolism. So a reaction in a closed system would eventually reach equilibrium. But remember, living things are not closed systems. So cells are not in equilibrium. They're open systems. Things are coming in, things are going out. So this is constantly changing. And again, metabolism as a whole is never at equilibrium. Maybe some reactions, some parts of it might be at any given time, but not in its totality. All right. That can be illustrated here. I love these diagrams from your book. These are great. So here's an example of a closed system in A. So here, think of like a hydroelectric power situation. Here's the little turbine here turning on the light bulb. So as water flows from the left to right, the turbine turns, the light bulb shines. But eventually, because there's no input, it will reach an equilibrium. And look, no capacity to do work, generate electricity in this example. But if we create an open system, which is more like a living thing, here's that constant input of water. Now we can continue to generate energy. So we don't ever reach that equilibrium, right? So that's why we can continue to do work. And then the last point here, which was a bullet on the previous slide at the bottom, I skipped over, is this idea that most metabolic processes, think back several slides, occur as multi-step processes. And so we see that here as well in our hydroelectric system example. Open system, so inflow of water, and then multiple steps. So we're able to draw energy out and do work at multiple places. So one of the ways that energy is moved around in living systems is through an intermediary, sort of an energy currency. And you've heard this idea before, that ATP is this energy currency of the cell. And so this connects those energy releasing reactions with the energy requiring reactions. They don't have to be side by side, but you can have this intermediary, and that a lot of times that's ATP. So cells do all kinds of work, chemical, transport, mechanical. So this is the kind of work that they do. And then they connect these exergonic and endergonic reactions often through ATP. Wow, remember, what's ATP? It's a nucleotide, adenosine triphosphate. It has that 5-carbon sugar, ribose, right? So you should know what kind of nucleotide that is specifically. And here's that energy cycle when we think about ATP. So ATP and water release a phosphate here. So there's a reaction, hydrolysis, breaks off one of those phosphates. That releases energy. That energy can be used to do work, build compounds, for example, in a cell. Your products from that are ADP, adenosine diphosphate, because you've lost that phosphate, but it's still out there. There's a phosphate pool in our cells. Those can be put together again to make more ATP. How do they do that? It requires some energy. You got to have an energy input. So that can be from those catabolic reactions. And so here's another way of looking at it. These exergonic reactions break things down, plug energy into ATP, and the endergonic ones use ATP in the energy from ATP to build bigger things. Now, if you look as well here, if you're actually building ATP from ADP in a phosphate, this itself is an endergonic reaction. So already the coupling of exergonic and endergonic is right here as well. So one of the themes that we'll see when we have these building reactions, the connecting these breaking down reactions and these building reactions, is we'll see a phosphorylated intermediate. You could think of ATP as almost being like that. You take ADP and phosphorylate it, add a phosphate. Now you have ATP. But really, when we're thinking about phosphorylated intermediates, it's something else is getting a phosphate added to it. So here's an example. So we want to go from glutamic acid and ammonia. We're going to combine those and make glutamine. So ATP phosphorylates glutamic acid. So here's the little phosphate here. Now in comes an ammonia group that can be tacked on to glutamine, and that's our end result. So here we see this in totality in panel B here. And pay attention, remember, we had an endergonic building reaction. We're putting together the ammonia and glutamic acid, but at the same time, we're breaking down ATP. So that's exergonic. And if we look at the numbers on this, the kilocalories per mole, we're still net losing energy, right? So where did that energy go? Think of loss as heat. Think back to entropy, that second law of thermodynamics. So this isn't some 100% efficient process. ATP can also drive mechanical work. So we saw how it can be important just for building things, putting stuff together through those phosphorylated intermediates, but it's also important just fueling cellular processes that we've already seen. So remember sodium-potassium pump, for example, or active transport in general. ATP fuels that. So ATP comes along. Actually, we do see phosphorylation occurring here, and this will pump whatever compound is out of the cell in this example. Or like we saw with cytoskeleton, how things move around with these kinesin proteins, ATP, for every little step this guy takes, that costs an ATP. Okay, so when ATP releases some energy, it also releases an inorganic phosphate. What happens to that inorganic phosphate in the cell? All right. Well, we just talked about it, right? It can be used to phosphorylate something else. One of your options here was it can be used to regenerate ATP, but the wording of that was that it's only used to regenerate ATP, and that's not true. It can be used for that, but it also often is tacked on to another compound, as we've seen. Let's talk about enzymes now. So enzymes are important because they speed up these metabolic reactions that we've been talking about. So enzymes are a catalyst. You probably heard that term before in chemistry class. And a catalyst is important that it may speed up a reaction, but it's not consumed by the reaction, like say the reactants are. And so it comes out the other end and it's still the same. And so an enzyme in living things is one of our macromolecules that acts as a catalyst. Most of the time, these are proteins. An important one that isn't is the ribosome. So ribosomal RNA is an enzyme, and that is not a protein. But for the most part, the catalysts in living things are proteins. So how does this work when they speed up these reactions? Well, here's an exergonic chemical reaction shown in this chart here. And we can see you don't just go from reactants to products in terms of the free energy. There is required some investment of energy to get the reaction going. And this is called activation energy. We abbreviate just EA. And so that's what you need to sort of overcome to get this rolling. And so what an enzyme does, or a catalyst does, is it lowers that activation energy. So in this example here, the blue line is uncatalyzed, so you need a greater energy barrier here. You'd have a greater energy barrier to overcome, whereas in the presence of a catalyst, you still need that energy investment, but it's not as much. And so that's why enzymes speed this up, because they're lowering that activation energy. All right, so summary of enzymes. What do? They lower activation energy. You already know that. They're not consumed by the reaction. Hopefully you remember that. Mostly they are protein. And when you see names like kinase, ATPase, DNA polymerase, that is often going to be referring to an enzyme because it ends in that ASE. So that's almost a sure sign that you're talking about an enzyme when you see that ending. Some enzymes don't have that ending, so it's not 100% matched up, but it can be a helpful clue. One more point to make on this slide is notice the delta G here. Even though we've added this idea of requiring this activation energy, that change from reactants to products, it doesn't matter whether there's an enzyme there or not. The delta G, that change in free energy, is no different whether the reaction is catalyzed or not, because you're still going from reactants to products wherever you're starting at with their free energy and ending at with the products. All right, so enzymes are typically very specific in terms of what they work on. So here's an example of an enzyme, and here's the thing that it's going to work on. That's called a substrate. So enzymes act on substrates, and substrates will bind to enzymes at a place called an active site. So that's what we see here. So this little substrate's going to go into this little nook in this enzyme, and when that happens, they'll form the enzyme substrate complex, pretty cleverly named, right? Hopefully easy to remember. And we can look at a couple of examples of what this might look like. Here's a cartoon example. The active site shown here kind of in pink. Here's the substrate binding. Here's more of a 3D model. You can kind of get a better sense of this fit here. So the shape of that substrate has to fit into that active site. You have to have the right polarity of the amino acids at that active site. So only certain things are going to fit there, and that enzyme therefore is going to be very specific. Now, keep in mind how proteins come together. You've got that amino acid chain, that primary structure that twists and folds and bends, and maybe multiple subunits come together. And so if you were just to look at an amino acid sequence, just one after the other of a protein, here's the amino end, here's the carboxyl end. The active site in its three-dimensional shape, so we see these purple amino acid residues making up the active site, they all come together in the 3D shape, but if you stretched out that protein, they might be at various places along that protein, but it's ultimately the three-dimensional shape that matters. All right, so another important factor here is when that enzyme substrate complex forms, it's not just this lock and key type of thing. So shape matters, the polarity of the active site relative to the substrate, all that stuff does matter, but then something else happens. So when you have that binding, the enzyme will actually change shape, and this creates what's called an induced fit. So the enzyme will fit even better to that substrate once it binds. So the fit is induced by the binding. And so this is going to influence or this is what's going to help catalyze whatever reaction we're talking about. So let's see if you got a sense of what induced fit is. So given these options, which of the following is correct? All right, so there were some choices there that might look pretty good and are important for that substrate enzyme binding, like the active site can actually fit the substrate, for example, but that's not about induced fit. The induced fit part is that option where we see the enzyme and therefore the active site actually changing shape once we have the substrate binding. All right, so let's look at that again and see what's happening here in this little cartoon. So here's an enzyme in its native state with nothing bound to it. When the substrate, in this case there's two of them, bind, the enzyme changes shape. And so this binding to the active site, what are some things that might be happening here? The substrates are oriented correctly, so things are sort of turned the right way. The enzyme might be putting strain on the bonds. So that's, if this thing's trying to break a bond or reorganize them, that's going to put some strain on it and accelerate that. Again, lower the activation energy, increase the likelihood of it happening. The substrate may need a certain microenvironment to go to that transition to the products. The enzyme might actually even covalently bond to the substrate, and that might be important for how it works. So these are all possibilities. All right, so continuing with our example here, our substrates bind, strain is put on chemical bonds. Maybe in this case, some atoms are brought in more close association as well. So some bonds break, some bonds form, and here we end up with two different products out of this. And then off they go. And here's our enzyme. Remember, the enzyme is not consumed by the reaction. It releases the products and off it goes. It can do this again. All right, so what can drive the rate of the reaction? Well, you could, in a test tube, add more substrate to a certain point, right? So the more substrate the enzymes have to work on, the more product you're going to get out of that. But it also depends on how much enzyme you have, because these are going to get saturated at some point. So if you had 10 enzymes, 10 of them are working, you got to wait, right, until they're done for the next substrate to be acted on. So once you reach saturation, the only way you could speed up the reaction is to add more enzyme. Now, in a living system, that might mean a cell needs to build more of that enzyme, crank out more of those proteins. In a test tube, that might mean, just pipetting some more into that reaction. So what else can affect the rate of reaction? Well, we can think about what might affect the enzyme directly. So temperature and pH can alter the shape and therefore function of a protein. So you heat something up too much, it might denature, the protein might come apart. Same with pH, too high, too low. And then there's all kinds of chemicals that can influence specific enzymes or generally influence enzymes. So let's talk about those. So here's the example of temperature and pH. And there isn't some universal, well, above a certain temperature, no enzymes work. Well, maybe above a very high temperature, that might be true. But in living things, there's a range in which enzymes will work. So here's a human enzyme, and it works pretty well. It's got a peak here, an optimal rate of reaction at about 37 degrees. That's human body temperature, right? But if you're a prokaryote that lives in a hot spring, if your enzymes work well at 37 degrees, that's not good for you because you do not live at 37 degrees. Your environment is much warmer. And this peak might be broader because these temperatures might fluctuate a bit more. But the optimal activity, or the greatest activity we see for this enzyme, is at 75 Celsius. And it does not work very well, or if at all, at 37 degrees. And likewise, our enzymes are not going to do great at 75 degrees. pH is another factor. So here are a couple of different enzymes. So pepsin, this is a stomach enzyme. Trypsin, this is found in your intestine. So those environments have different pH. Your stomach has a fairly low pH, very acidic pH. And those enzymes that are found in there work well at those pHs because that's where they have to live and do their work. But trypsin, which is in your small intestine, this does much better at slightly higher pH, a little more than 7, somewhere close to 8. And it will not work very well. See that you see no red line here down by below 5 here in pH. So pH and temperature, both important. And I would also emphasize, notice there's a range, right? So there's often a window of where an enzyme can work. You wouldn't necessarily want it to be black and white, like this only works at pH 2, because if pH 2.1, that's not so good. So there's a bit of there's some shoulders here to these optimal activities. All right, so let's talk about these other compounds. So some enzymes need something else to bind to them to actually work, and these are called cofactors. So it's a factor that's required for the enzyme to function. And so these are non-proteinaceous, and they may be organic or inorganic. So inorganic, think metal ions. Think of the product label you might see on a food that you're eating or your multivitamin. Why do I have to get, some minuscule amount of magnesium in my diet every day? Because that magnesium is a metal ion and it's an important cofactor for some enzymes. They won't work if you don't have it. You don't need much of it, but it is essential. Specifically organic cofactors are known as coenzymes, but really they're just like the magnesium thing. They just need to bind to the enzyme for it to work. And that may be temporary. It's not necessarily a permanent association with the enzyme. And we'll see coenzymes happening, cropping up when we get into more detail on metabolism. Now, enzymes can also be impacted by inhibitors and activators. Now, just here are some of the examples here. So here's an example of substrate binding to its active site. Deep fits here, perfect, off they go. Here is an inhibitor. This is this dark blue substrate. You can think of it like that. But the enzyme's not really acting on it. It's just blocking the active site. So when along comes the normal substrate, it can't go in there because this thing is in the way. So this is inhibiting the enzyme. So this isn't always just some bad thing. This may be a way to regulate enzyme function through inhibition. So this is competitive because the inhibitor and the substrate are competing for the active site. In non-competitive inhibition, the active site is not blocked. So look, it looks wide open. What's the problem here? Well, this inhibitor is bound somewhere else at another site. This is called an allosteric site. Allo means elsewhere. And it's changed the shape of the enzyme. And so much so that the active site can no longer recognize the substrate. So it's not blocking it directly, but it has the same effect. It's inhibited this enzyme. Okay, here's a question for you. HIV protease. So this is an enzyme found in the virus HIV, human immunodeficiency virus. And the amino acids at the active site of HIV, so in here, active site, are highly hydrophobic. So you're working for a pharmaceutical company designing a drug that's going to be an inhibitor of this active site. What kind of compound would you want to synthesize to act as an inhibitor here? Right, so hydrophobic. So the active site has hydrophobic amino acids. Therefore, you'd want an inhibitor that's hydrophobic as well. And I'd say the other options in that question, they're all basically a similar theme on something that's polar. So the odd man out there was hydrophobic. So let's add the layer here of an activator. And we're going to use this example here of this protein this is its active form here. And it can go from being active to inactive. So here, that's what we're seeing here, this oscillation between active and inactive. And then this may be somewhat unstable. So this is kind of a dynamic process going between active and inactive. But this inhibitor in this example is binding, and that's really stabilizing that inactive form. So it's like, yeah, you're going to stay shut off for a while until that inhibitor is gone. So that stabilizes the inactive form. The other example here is this allosteric. Remember, allosteric is something that's binding that's not the active site. This is binding somewhere on this protein that stabilizes the active form. So this is helping the protein basically stay switched on, and hence it's called an activator. Something else that we see here is this idea of cooperativity. And this is kind of cool because here we see this is a multi-subunit protein, an enzyme. So it has multiple active sites. And what we see here is when one substrate binds, it stabilizes the form, just like we just saw with the activator stabilizing the active form. But here it's just the substrate. It binds, it stabilizes the whole thing, and now all these other active sites are stable and say ready to go and bind a substrate. So this is called cooperativity. The binding of one stabilizes the rest, and now this enzyme is going to work more efficiently. All right, so remember, back to slide 2, biochemical pathways are often multi-step and involve multiple enzymes. And I mentioned how that's important because that allows for multiple places for the pathways to be regulated. And so we can see that here. So here's initial substrate. It goes through a couple of intermediates, these different enzymes, and here's the end product. Interestingly, this end product, boink, is an inhibitor of this first step. So when you get a lot of end product, it's like, whoa, we don't need any more of this. This itself will switch off this first enzyme in the process. And, when this starts to run out, this will stop binding because there's not very much of it, and this pathway will start again. This is called feedback inhibition. So a product or intermediate even acts as an inhibitor for an earlier step in the process, and in this case, grinding it to a halt. Now you can have positive and negative feedback on a reaction or a biochemical pathway. So here's just showing that same idea. Here we see isoleucine. So we're taking threonine, that's an amino acid. We're converting it to isoleucine through these multiple steps. When you have a lot of isoleucine, it will bind to this enzyme and prevent it from working, this first enzyme. So it's an allosteric binder. But when you have less isoleucine, guess what? That allosteric site is open, the active site is available, and this pathway will proceed. All right, that's the end of this chapter. See you next time.